Rihanna Luna, and today is November 18th, 2018, and I'm going to be going over the second experiment for week 3. This involves gram staining some slides. I just finished the first experiment, and the first exercise included using sterile swabs again to swab the inside of my mouth, my teeth and gums. And in the first slide, we had Graham stained it with a crystal violet dye, and we put the specimen or the bacteria onto the slide. Then we heat fixed it by lighting a candle. I'll show you here. So we had the candle lit, and we actually used the clamps here, and we would hold the slide. You would pick up the slide and you would just heat fix it by waving it back and forth 5 times. And then you would let the slide sit for about 10 minutes. After the 10 minutes, we put the slide inside this little container to actually stain it. And when we stained it, we let it sit for another 10 minutes to make sure that the stain sits and it's not easily washable. After the stain has sat, we use tap water to wash off the color from the slide. We tap dry it with the napkin so we're not rubbing it and taking off any possible cells from the slide. And once the slide is completely dry, we then put it on the microscope. So here I have my first slide that I did, and I already viewed and took pictures to include in my lab report. The second slide is sitting right now drying. I'll explain that process. We used Congo red, and so first step was to actually drop Congo red onto the slide. Then we took the sterile swab and got the cells from my gums and my teeth. Then I placed the swab onto the Congo red, and after the swab sat into the Congo red, displacing the cells into the drop of Congo red, we took another slide, which is right here, and we slid it from top to bottom of the slide at a 30-degree angle, spreading all the cells across the slide. So now it's air drying right now. I'm going to take a look at it on the microscope, and we're going to examine the cells in the Congo red dye, and then I'll take a picture and put it in my lab report. The second part of the exercise includes using some microbes that we grew in broth last week. We're going to be using epidermis, Cerevisiae, and yeast. We will be putting those specimens on the slide by using the loop. We're going to heat fix them, doing the same process. We're going to gram stain them. We're actually going to use, though, some PVP iodine gram stain as a second step. It's right here. Then we're actually going to use a decolorizer, which is this solution here, for all 3 slides. So that requires an additional 60 seconds, 30 seconds in between each step, but that process will help differentiate bringing out the cells on the slide. So I'm really looking forward into viewing and comparing each bacteria, and I'll include those pictures in my lab report. So that includes the second portion of this exercise, and everything that I do will be included, like I said, in the lab report to view from what I've seen on my microscope. Okay, thank you.